Hi everyone, welcome to the first of two of the project discussions for the available projects in my lab. So I pulled these presentations together pretty quickly, so this isn't going to be super polished, but I'm hoping to give you a sense of kind of the overviews of some of these projects and then some of the sub-projects that each of you will get to pick from when you get done with all of your training. So the first of these is going to be the longest video because we have the greatest amount of data and the greatest amount of need for students to work on this project. This is where my current funding comes from, so I'm hoping most of you will be intrigued by this project enough to want to work on it. And really the goal of the project is to identify molecules that may be mediating an interaction in a couple of human pathogens. All right, so let's get started. I first need to explain what biofilms are. If you've already taken my class, you should already know this, or perhaps a review is in order. But microbes in general in nature, particularly bacteria and archaea, tend to exist not as individual cells floating around or crawling across surfaces, but rather they tend to form these multi-species communities that are attached to surfaces. And these are called biofilms. Biofilms are found incredibly commonly in many natural environments, mostly aquatic environments or partially aquatic environments. And these are where bacteria can get together, produce this sticky matrix of stuff, and all exist attached to the surface of rocks or plants or other things. And they do this because letting nutrients to come to you through aqueous solutions is far more efficient than expending the energy to try and swim or crawl around by yourself. You also get to take advantage of community dynamics. So if other people or other bacteria in your biofilm produce food that you can eat and they do it as a waste product, then you can live in harmony. But there is competition for nutrients and resources. We also know that nutrients in the environment, like minerals and sugars, can stick to surfaces often, like in the pores of rocks or wood. And so this is a good place for microbes to settle down and form these biofilms. Now these biofilms themselves, like I said, are made of the cells themselves and the sticky matrix. And the sticky matrix is actually, you can see it in these scanning EM images. And this top one is false colored. But you can see that this matrix is actually made out of polymerized sugars, polysaccharides, extracellular DNA, and proteins that are out there assembling all of this. And this matrix is really important because it can protect against attacks. When there's predators that come in and try and eat you, you can hide inside this matrix. It prevents certain types of chemicals from penetrating into it, like antibiotics. But it can also help trap nutrients as they flow by. It also gives you that stable structure, if you're the microbe, to stick to that surface. So they're very important. And there's 5 known stages for biofilm formation, right? And this has been particularly well studied in pathogens like Pseudomonas aeruginosa, but it is now known to be a common feature amongst almost all bacteria on the planet. So the first of the stages is a reversible attachment, and this is when aquatic microbes are swimming around using their little tails called flagella, or flagellum, and they sit down on a surface and sense that they are on a solid surface. They attach to this surface reversibly using other structures like type 4 pili and fimbriae, which are extracellular sticks that stick out from the surface and help convey the message inside the cell that there's a solid surface. Cells then go through irreversible attachment when they start secreting this polysaccharide. And as the name implies, at this stage, they're kind of locked into the biofilm mode of growth. They shed their flagella and their other extracellular structures and just start using all the carbon that they can to pump out this, the matrix. This matrix is also called extracellular polysaccharide or extracellular polymeric substance, otherwise known as EPS. As the cells start to multiply on the surface, they produce more polysaccharide mix or matrix. Other cells can get recruited or sit down and join the party. And this will continue to grow through stages 3 and 4, which are known as maturation 1 or 2. Now you'll notice that for the most part, for some of the better studied ones, they get these large globules that stick up into the air, and these form all over the surface. It's not generally one giant thick mat. And the reason for this is because you have to allow water channels in these structures to help bring in additional nutrients and oxygen. Step 5 is that when the biofilm is fully mature, some of the cells will start expressing their swimming proteins, and then these will leave the mature biofilm to go form biofilms elsewhere and to spread. So biofilms are very important. We've known these structures have been around for billions of years. There's evidence of these in the fossil records, and we have evidence of current day fossils that are still found off the coasts of Australia and other types of aquatic environments. They're environmentally very important, so they help contribute to the global photosynthesis chain, as you can see with the green algae on the rocks. And algae are not bacteria, they are eukaryotes, but we also have photosynthetic bacteria that participate in these types of communities. They also are primary producers, so they're very important for the food chain because a lot of organisms graze on them. They're also environmentally important because interactions between bacteria and archaea and plant roots are also very important for the health and survival of a lot of our plants, which of course most of us upper organisms rely on for food and nutrition. So here's a scanning EM of a plant root covered with bacteria. Industrially, they're also important. They can contribute to biofouling of tubes where they clog up, if you've ever seen a gross shower drain. But in industrial complexes, these can be also even more of a problem because they can contribute to blockage of pipes. They can form on the surface of fruits and vegetables. So this is, I think, a cantaloupe skin, if I remember right. They can cause foodborne outbreaks. They can also colonize the bottom of ships that sail in the ocean and lead to degradation of the metals. Most importantly for us probably are the medically important ones. So biofilms are very important medically, either from the dental standpoint, as plaque on your teeth is actually bacterial biofilms. Also, in interactions inside the human body with eukaryotic cells, biofilms forming on the surface of cells can lead to disease in some cases. Biofilms are also really important for indwelling devices like catheters because bacteria can readily colonize the surface of plastics. And the problem here is that the biofilms are incredibly medically resistant to antibiotics or to physical disruption and removal. So they can be contributing to major systemic diseases. They're also, by the nature of the matrix, very resistant to immune cells, which will come in to try and phagocytose or destroy these bacteria. But bacteria in these communities can also produce and secrete toxins that will kill things like neutrophils and other immune cells. So they're very becoming much more important as antibiotic resistance is on the rise as well. So we study medically important bacteria that also live in many different natural, non-human associated environments. And I chose a long time ago to start studying organisms that actually interact with each other. And I want to study these interactions and how they impact human disease. And so one of the best model systems for this right now is in a disease in humans called cystic fibrosis. Cystic fibrosis is a disease that is caused by lots of different types of mutations in a single human gene called the CFTR gene. This gene encodes a regulatory chloride transporter, and it is expressed in lungs and the gastrointestinal tract. The results of these mutations can lead to some pretty severe diseases, But the disease itself doesn't typically kill the patients or kill the individuals with the disease at early on in their life. It can, but it doesn't often. So the lifespan of people with cystic fibrosis in the U.S. right now is about 40 years old. So they have plenty of time to have children. And if you'll notice in this infographic here, that in the U.S. right now we have about 30,000 people living with CF. In other parts of the world, it is more rare. It is found far more commonly in Caucasian populations, where 1 in 4 people actually carry a defective version of this gene. And even the heterozygous people who have one normal copy of the CFTR gene and an impacted CFTR or mutant CFTR gene can have symptoms of the disease. Most of the time, CF is now diagnosed in newborn screening or in prenatal genetic testing. And the mutations work like a standard mutation. But the symptoms of cystic fibrosis include a massive, thick mucus that builds up in the respiratory tract, particularly the lungs, and there are digestive issues that go along because of the expression of this disease in the gastrointestinal tract. People with cystic fibrosis, if diagnosed early, are seen quite frequently in CF clinics. We have one pediatric CF clinic here in Louisville. We have an adult CF clinic here in Louisville. There can also be reproductive problems and an assortment of other types of diseases that go along with this, including poor nutrient absorption and immune issues. So part of this disease actually impacts one of the cellular processes called autophagy, which is how the cells kind of recycle garbage inside the cell. And so when you have that, you have immune function defects as well. There is no cure right now for CF. They are working on gene therapies for this. There are a couple of new medicines that have come out in the last couple years that reduce the number of symptoms, but generally are for people with only very specific mutations. So they reverse the effects of some of the mutations, but not the major ones, right? And even in the reversed people or people that are on some of these drugs, they can still get bacterial infections. And the reason for this is because when you have a defect in the chloride ion transporter expressed in the lungs, it causes a dehydration outside the cell because the chloride ions and thus water don't flow outside the cell. And therefore the mucus that normally is well hydrated and can be removed by coughing or by ciliary beating in the upper respiratory tract cannot be accomplished in people with CF. And you get this thick buildup of what we call mucus or sputum. And mucus is actually made up of a lot of sugars and salts and amino acids, right? So it is a very carbon-rich source of food that is well loved by certain bacterial pathogens. So bacteria can get in there and they can eat all of these carbon sources and colonize the respiratory tract. And of course, when in doing so, they will also secrete virulence factors that can damage the host lung, cause pneumonia, and in worst case scenarios can cause septicemia and sepsis and transient bacteremia. But sepsis is the big problem because some of these organisms that can colonize the lungs are very antibiotic resistant naturally because of their lifestyle outside of the host. So what species do we commonly see in the CF lung? Well, it turns out the CF Foundation, which is a nonprofit here in the U.S., tracks all 30,000 people in the U.S. through their doctors by offering CF doctors compensation, offering clinics compensation to report back data. And every year the CF Foundation comes out with an annual patient registry report. And the last one came out in 2022 because it takes them a little while to get the data. And what you can see is that there are certain microbes that are found the most common, and these include things like Pseudomonas aeruginosa, which tends to be low in children and gets higher over the course of their life, so more common in adults. Things like Staph aureus are primarily diseases of children with CF, and you can see it infects a lot. Now, Staph aureus doesn't tend to kill kids with CF quite as much, but can cause long-term bouts of pneumonia. And we also know that Staph aureus is becoming more and more antibiotic resistant. Perhaps you've heard of MRSA, which they track directly. This is methicillin-resistant Staph aureus, which has been kind of steady and maybe even going down in the population. And I'll show that in a minute. Some other ones you may not have heard of would be like things like Haemophilus influenza, which is also a childhood-associated CF pathogen. Stenotrophomonas multifida, shown in the purple line, is fairly low, but is still one of the top 6 pathogens in the CF, along with the Burkholderia cepacia complex and Achromobacter xylosooxidans. We're going to be focusing on a couple of these pathogens today, but several of these you'll hear about in a little bit. The other thing I want to point out is that the commonality, or the most common pathogens, actually has changed over the last 20 or 30 years. It used to be that Pseudomonas aeruginosa was the most common pathogen, and then about 2002 this kind of started settling down as we generated new therapies for Pseudomonas aeruginosa. And Staph aureus now has become the #1 infectious agent across all CF people with CF. And we shouldn't call them CF patients because they're people living with CF. So just be conscious when you're speaking about CF that you don't just call them patients, because most of them are not patients most of the time. People with CF, traditionally, if they've been diagnosed and stick to the plan, will go visit a CF clinic once a quarter every year, so 4 times a year, unless they're having pneumonia-like symptoms, where they will go in more frequently, and their lung function is low enough, they will have what's called an exacerbation event and may have to be hospitalized for treatment. You'll also see that things like MRSA are creeping up until about 2019, and they started dropping. And other ones, these lower ones, are fairly consistent across the years. So these are the top 6: Staph, Pseudomonas, H. influ, multifida, xylosooxidans, and the Burkholderia cepacia complex. And you might be wondering why they call it the cepacia complex. And that's because this is a group of 21 or more organisms that are all very closely related, that used to be called Pseudomonas cepacia, even though they're not genetically that closely related. And now, as more work has been done, we have split these off into the different species, but they all contribute to CF pathogenicity. We also know that antibiotic resistance isolates coming out of CF lungs are becoming more common. And this we can see with Pseudomonas aeruginosa, and you can see the little red line, which is the multi-drug resistant Pseudomonas isolates, are becoming more prevalent in adults. Still not eclipsing the non-drug resistant Pseudomonas. I mean, Pseudomonas itself is naturally drug resistant to certain antibiotics, but now we're seeing even more resistant isolates popping up. And the same holds true for methicillin-sensitive Staph aureus and methicillin-resistant Staph aureus. So there is an urgent need to try and find new drugs that can help with some of these pathogens, particularly for Staph. Staph is a much larger pathogen in the global community outside of CF, as it causes many hospital-acquired infections. Pseudomonas tends to be a pathogen that's really known in the burn wards, so people that have been badly burned, as it's the #1 pathogen in there. But Staph is certainly a huge pathogen that kills a lot of people every year. So just to sum up some of this, what we see is there's some ecological trends in what we culture out of CF lungs. And these are things like Staph aureus and H. influenza tend to be childhood associated, though this is kind of spanning and becoming more prevalent in older adults. Pseudomonas aeruginosa is found mostly in adults in their infections. What I find the most interesting when I went up to Harvard to study some of these pathogens is that there's these end-stage pathogens, which are the ones that are a little bit less common, like Burkholderia cenocepacia and other Burkholderia cepacia complex members, Stenotrophomonas multifida, and Xylosa oxidans, Achromobacter. When these come in, they can actually wipe out everything else and turn the lung into a monoculture. So for the most part, people with CF, with all this mucus, they have lots of things growing in there, right? But when these end stage ones come in, they come in and they just take over the entire community and kill everything else. And then they kill the patient, which is not a good thing. But it presents a really interesting question of how do they do that? How do they come in and wipe everything else out? So I just want to point out also that the CF Foundation, once again, it tracks all these people. And so they have done this since the 1980s. You can actually request data from them. So well, let me tell you what they collect. They collect information about each CF patient, like their age, where they're living, what hospital they are attending, what their CF clinic is, their gender, their sex, their whatever other demographic, race kind of information. The health status, so they look at the BMI, their weight, their lung function, which is the forced expiratory volume in 1 second, or FEV1, along with some other respiratory metrics. Their height, whether they're pregnant, whether they're having any mental illnesses at the time, their diabetes status. They also collect lifestyle info, like marital status, whether they smoke or they're not, or they're around smoke, what kind of job they have, if they have a job, whether they have insurance or not. And then CF Foundation will collect information about the microbial profile of the sputum. So when each person goes in, even if they're healthy, they produce a lot of mucus. They're asked to cough into a cup and cough some sputum into the cup, and this gets sent to microbiology labs. Now, different microbiology labs in the hospitals across the U.S. test for different pathogens, and they have different procedures, so this isn't always consistent. But the CF Foundation collects the data for over 90 different species, if they're tested for. And then whatever treatments these CF patients have going on at that particular visit, so whether they're on preventative inhalers, whether they're taking antibiotics, whether they have stuff to stimulate the coughing or to loosen the sputum, whether they're on their the machines that shake their body to induce coughing and loosen stuff up. So they collect all of this. So I actually, a long time ago, requested some data, and they sent me, I requested data for a number of years. I requested a lot of different types of data, and they sent me 1.1 million records back. Now, this data is a little dated at this point, but you can see that, in 2015, which was now 9 years ago, they saw 162,000 people. They saw 162,000 clinic visits, many of them for, multiples for one of the people in the country. And we could actually take this data and say, do we see evidence of pathogens getting along or fighting or ignoring each other? So I'm going to show you, this is a statistical analysis of the data sets. And we basically asked, are there any pairs of CF pathogens that are more or less likely to co-occur? And you couldn't just ask this using raw data, because some of our pathogens are far more prevalent than others. So you have to ask, well, given their overall prevalence in the population, when these things are found together in the lung, is that more commonly expected compared to random chance, or less commonly or equal to random chance? So here we have this box plot where we have pairwise comparisons. So this top red one shows the Candida and Aspergillus. And these occurred, these are both fungi actually. So these were found more commonly than expected from random chance over the 7 years that we analyzed. And what I want to point out down here at the bottom is the Stenotrophomonas, the Burkholderia cepacia complex, aeruginosa, influenza, and aureus. And these are 5 of the big 6. I didn't request a Achromobacter data. I don't know why. Maybe it wasn't on their spreadsheet at the time. And what you can see is many of these are having blue boxes between them. So if you follow the boxes, the columns and rows, and this would imply that these things are found less than would be expected by random chance, and thus they might be fighting, right? And there are 2 red boxes within this region, though. So down here is a red box for where Stenotrophomonas and Staph aureus actually, when they're found together, it's more commonly than expected by random chance. And then H. influenza and Staph aureus. And we know that H. influenza and Staph aureus is a true thing because they're both commonly found in childhood, children with CF. The Stenotrophomonas and Staph aureus one was kind of a surprise, but we care a little bit less about whether they're getting along than whether they're fighting. Because if they're fighting, then perhaps they're producing things that kill each other and that can be developed into antibiotics. So we had this data in hand, and we actually went into the lab, and a former undergraduate in the lab developed a method for us to look at these pairwise interactions and see if what we see in the human data set can be recapitulated in the lab. To do this, we grow an organism A and an organism B, and then the morning of the experiment, we mix these together in equal proportion. We then put these into 96 well plastic plates and let them develop over time, and the biofilms will form on the bottom of these PVC plastic plates. So we incubate these in the incubator. We put some gas permeable film on there, and then when we want to quantitate how many organisms are in there, we can actually peel off that rayon film, and we actually put our biofilms in a sonicating water bath. And the act of sonication is using frequencies to shake at a very, very small scale these cells, and that is sufficient to release the cells from the biofilms. We can then serial dilute our stocks because these are quite concentrated, and we can plate these. And we do this through serial dilutions like you do in chemistry class and drip plating, which means we take 10 microliters of 8 different dilutions for a sample and plate it down the surface of a square plate. Now this plate here shows a monoculture. So if we're only looking at one organism, then you can count specific lanes and calculate back the concentration of the microbe in the sample. When we have co-cultures, this becomes a little bit more complicated, but luckily for us, our pathogens all look different on these media. So for example, this upper one over here shows Stenotrophomonas, which is this large, quick-growing yellow colony, and Burkholderia cenocepacia, which are these tiny pinpoint-like dots. Similarly, down here at the bottom, Staph aureus are these large white circular colonies, and Burkholderia are the smaller, slower-growing ones. So if you once you get to know what these things look like, they become easier to quantitate, and you can quantitate them all together in drip dilutions. So with this in mind, Rachel Thompson, the former student, did pairwise interaction studies to look at these. And the data that we're going to be looking at is going to look like these bars here, where we have 2 different days. So we analyzed biofilm formation 1 day after inoculating or 3 days after inoculating. And for this particular organism, we're going to look at the amount of Stenotrophomonas multifilia when it was in these different conditions. And in this case, the green bar is the monoculture, or this organism by itself. So this is how much it grew just on its own. You can see when we mixed it with these other organisms, whether it's the red bars that are Achromobacter, the orange bars that are Pseudomonas, the blue that's Staph, or the purple that's Cenocepacia, the levels of Stenotrophomonas look very similar to monoculture. Stenotrophomonas tends to ignore everybody. It does fine on its own. It does fine when it's in the presence of anybody else. It is not inhibited in any way. In contrast, the other gram-negatives that we study, which are Achromobacter, Pseudomonas, and Burkholderia, you can see that for each of these, like Achromobacter, when it's by itself, are the red bars in the lower left-hand panel, and when it's with these other ones, you can see the relative amounts there. What you don't see is green bars, and what that means is Achromobacter is killed by Stenotrophomonas. Similarly, we see the same in Pseudomonas aeruginosa. Stenotrophomonas tends to kill that one as well. It also doesn't do so good with staph, all right? And then the Burkholderia tends to grow well on its own and with others, except for Stenotrophomonas. The last graph I'm going to put up here is actually the basis for the project, and that is the Staph aureus. So Staph aureus is a little bit unusual amongst these in that it's a gram-positive, it's a coccus, it's not rod-shaped, and it's a very, very fast grower. So at day 1, you can see the blue bars are staph when it's growing by itself, and it has some robust growth. And I should have pointed out that on the axis, the y-axis over here, this is CFUs per mil. CFU is a colony-forming unit, and it represents one viable cell that sat down on the surface of the plate, grew up sufficiently to form a countable colony. So it's like one living bacteria per mil from the original stock. So it's a concentration of bacteria. And staph does fine at day 1, but by day 3, it's actually wiped out by a fair number of stuff. It's not wiped out by itself, and it actually likes Stenotrophomonas. Now that's important because in the CF data from humans, we also saw that these two organisms were found more frequently than be expected by random chance. It was one of those 2 red boxes that we just saw. We also know that Pseudomonas aeruginosa, which would be represented, Pseudomonas, actually produces 3 different anti-staphylococcals, so that that is probably the reason it is not, we don't see staph when it is grown in the presence of Pseudomonas. The Achromobacter and the cenocepacia were quite surprising because Achromobacter and Burkholderia cenocepacia are very slow growing organisms, and staph is a very fast growing one. So this doesn't seem to be caused by nutrient acquisition or, sequestration. And that would imply that it is actually doing something very specific. I'll say right now that we had a student try to follow up on the Stenotrophomonas stuff. We weren't able to track down anything really very well, so we didn't follow up on that. But we had a student who did this work who wanted to follow up on this lack of a purple bar here. So staph seems to be killed by Burkholderia cenocepacia, and that is once again surprising given their relative growth rates. So the whole project is going to be based on finding out how this works. All right, so we have some data to show a little bit more information about this. So if you take and try and co-inoculate these organisms, which is what we did in the previous one, and in this time we're putting an equal number of cells, we can see that Burkholderia, when it is a monoculture, shown here in red on the left-hand panel, and it grows to very robust levels or concentrations by 1, 3, or 7 days of biofilm formation. So it's doing fine on its own. When you actually take Burkholderia cenocepacia and you mix it with staph and you count the Burkholderia that remain, once again, it does great. It's the green bars or the green lines here on the graph. Staph aureus, when it's by itself or monoculture, is shown in the blue line. It does really well over time as well. But Staph aureus, when it is grown in the presence of cenocepacia, after 1 day, so by 3 days, it has become not detectable, or ND. And this is statistically significant. So this is when you inoculate them both at the same time. We also wanted to ask, if you have an existing biofilm of Staph aureus, can Burkholderia cenocepacia wipe that out? And the same holds true over here, is that we see significant reductions in the amount of staph, but it is not completely wiped out like we see when we co-inoculate these. But we're looking at a 2 to 3 log loss, so 100 to 1,000-fold loss in Staph aureus, and that could be clinically significant. We can also look at how these things interact using other techniques besides viable cell counts, which we will come back to in a little bit as we explore this interaction more. But I want to point out that a couple of things. We actually were able to use genetic modifications to turn organisms different fluorescent colors. So we have a green fluorescent Staph aureus that we obtained from a collaborator, and we were able to put in a red fluorescent protein into Burkholderia cenocepacia. When you grow these things together, on day 1, you don't see a ton of Burkholderia and you see a lot of staph. And these are biofilms. And it turns out Staph aureus, its biofilm doesn't have these magnificent mushroom kind of balls in the air. It has like a flat blanket, a big fuzzy blanket of biofilm. Now I'm going to show, so this is confocal laser scanning microscopy, which allows us to look in 3 dimensions at our biofilms. But these are just single slices. Within, like in the middle of the biofilm. By day 3, Staph aureus by itself is getting thicker and thicker, brighter and brighter. Burkholderia cenocepacia by itself is forming these, starting to build some of its mushrooms. So it's actually one of these that builds the large towers. When you slice into that, you can see the rings that form the stalks of these. But when they're together, they're doing slightly something different, where it looks like there's clusters of Staph aureus, and these are surrounded by the red Burkholderia cenocepacia. There's another image of that. You can see there's this huge, big, diffuse pocket of Staph aureus surrounded by cenocepacia. And by day 7, you have this dense mat of Staph aureus on its own. The Burkholderia cenocepacia have these large clusters of water channels and mushroom shapes. And then once again together, you actually have this really weird pattern when they're together, where the blanket isn't, like the Staph aureus is kind of clumped up, and the Burks are growing on top of that. And you can actually do movies of this. So because laser confocal scanning microscopy allows you to image in different depths automatically, you can actually build 3-dimensional images. So this first video up here on the left is going to show as we layer on images on top of each other. This is what the 3-dimensional image of the biofilm looks like of these two together. Oops, sorry. So here's the bottom of the biofilm. We're getting to the middle part of the biofilm, and you can see these punctate pockets of Staph aureus covered by the Burks. And if we take some of these, we can actually take these images and we can flip them around. So here's the top side of the biofilm in the lower hand right video. And if we take this and we can manipulate it so that we can flip it upside down. And what it looks like is that there's these pockets of Staph aureus that are have the Burks on top. So the way I think about this is that the staph are very, very afraid. They cluster together, they huddle together for protection. And then the Burkholderia are coming in like zombie style, jumping on the backs of these staph and starting to do something to them to start killing them. So it's pretty cool, pretty interesting. The other thing we have to think about is that, you know, bacteria, when they grow in an environment, can acquire lots of different genetic elements depending on what that particular cell runs into, what other cells it runs into, what other defenses it's picked up. It's a lot like human beings, where we have a species level, but each of us have different immune responses, right? So it turns out that's the case in bacteria as well. So we've been using a single clinical isolate of Burkholderia cenocepacia and testing it against a single clinical isolate of Staph aureus. And then the question is this effect conserved across both species? So we first asked, is this particular isolate, which is an epidemic isolate from CF clinics, is it inhibitory to other Staph aureus clinical strains? And we got a bunch of strains from Norton Hospital here in town and other sources from like Emory University, where I have a collaborator. And we looked at methicillin sensitive strains and methicillin resistant strains. And you can see that when they're by themselves, they grow fairly robustly. And when they're in the presence of this particular cenocepacia, most of them are reduced, very high levels, regardless of whether they're MISA or MRSA. The MRSA tend to be a little less killed than the Missus strains that we tested. So that's kind of an interesting little thing. We flipped this around and said, can other Burkholderia strains kill Staph aureus? So here we're looking at the growth. Oh, hold on. The other thing we looked at is whether other gram-positives could be killed by Burkholderia cenocepacia J2315. And it turns out most of them, so we tested other staph strains, Enterococcus strains, Bacillus strains, and Listeria monocytogenes. And in fact, most of these are not killed by Burkholderia cenocepacia. There's a couple of exceptions. Staph epidermidis is relatively closely related to Staph aureus. It too was killed, but not other staph species. And Listeria as well was knocked down about tenfold, though it was significantly different. Some of these others, you can see that the crosshatch bar, which is the co-culture conditions with Burkholderia cenocepacia, are actually higher than their monoculture or clear bar counterparts. So then once again, we flipped this on the head and said, can other cenocepacia and other Burkholderia species do this to staph? And so here we have staph counts. We have the medium alone, so staph by itself grows to a very high level. All of the members of the cenocepacia species that we tested, which are both environmental and clinical isolates, can completely reduce Staph aureus over time. But not the closely related, well, to a lesser extent, the closely related other Burkholderia species that are either in the complex, which include the Orbicola dolosa multivorin strains that we tested, and even just slightly outside the complex, Burkholderia pseudomallei. So these are all strongly reduced, but they're not eliminated like we see for cenocepacia. So we asked then, can Burkholderia cenocepacia produce and secrete a compound that can kill Staph aureus? In other words, is this inhibition of Staph aureus cell-to-cell contact mediated, or is it something that is diffused away from the Burkholderia and that can kill Staph aureus? The reason we ask this question, because antibiotics that we traditionally think of antibacterials in the clinics, like ampicillin or amoxicillin, tetracyclines, these are all small metabolites that are made by one cell to kill other competitor cells and are small molecules that are diffused away, right? So they're not based on cell-to-cell contact. So this is what we would want to see if we want to develop a new antibiotic in general. So what we did is we grew up Burkholderia on its own or Staph aureus on its own in biofilms for 3 to 7 days. We then removed the supernatant, took just the biofilm cells, lifted those with sonication. We took the supernatants off the top of the biofilms, spun them down to remove cells, then we filter sterilized to get rid of any remaining cells, and we just had the supernatants. And with this, you can see that here's Staph aureus counts on the left-hand side in just growth medium called tryptic soy broth, right? It grows to very high levels by 1, 3, or 7 days into biofilm formation. If we mix it with its own supernatant, that's shown in the dark blue bars here, it grows pretty well. It's a little depleted, and this makes sense because Staph aureus had already been grown in a media, used up all the available carbon sources. So this is kind of like a nutrient poor medium that we put it in. And that's why you see significant differences here, but it still grows up to 7 days, or it survives up to 7 days post-inoculation. When we take the supernatant from Burkholderia grown by itself in biofilms, we can see that we do not detect any Staph aureus growth. This suggests that there's a secreted compound that Burkholderia produces during biofilm growth all on its own that is sufficient to kill Staph aureus. We compared this then to looking at other types of cells, so whether it's Burkholderia overnight cultured cells, the supernatant from cultured cells that were just overnight. These would be planktonic, non-biofilm-like cells. Or if we take Burkholderia cells and we kill them with heat or sonication, or if we concentrate them. Like all of these produce, most of these produce a little bit of a reduction, not even tenfold reduction, and none of them produce such a strong inhibition as the the biofilm supernatants. All right. So another question we had is it actually killing Staph aureus, or is it just preventing biofilm formation? So we used a couple of ways to quantitate this. We used confocal microscopy to look at the actual cells, and then we used quantitative analysis of the fluorescent proteins and total proteins in order to assess whether there's any cells in there to begin with, in either the biofilm itself or the supernatant on top of the biofilm, which would be considered planktonic. And this gets a little hairy, and if you don't understand this, this is okay. But what we're looking at is each of these panels in microscopy are shown quantitated over on the left-hand side. So if we just grow Staph aureus in a biofilm in a nutrient-rich medium like LB, you can see the green staph here. There's lots of them. By day 3, you see even more. The focal plane is a little harder to tell. You have all these like darker dots because that's other staph that are just out of frame. And there's also Burkholderia cells in here. Is that true? I don't remember what I did. Any case, you can see the quantitation of these over here. So total protein, hold on. Oh yeah, you have to, oh, yes. The red cells are dead cells, sorry. There's so the green is living Staph aureus cells. The red is actually not Burkholderia cenocepacia. It's a dye we use that only shows up in cells with compromised cell walls. So it shows dead cells, right? So on day 1, you have lots of living Staph aureus. By day 3, some of those have started to die. And this is the biofilm cells and the floating planktonic cells on top of that. When you quantitate this, you can see that those have pretty robust measurements for total protein. There's much more living cells in the biofilm on day 1. They're more in the planktonic by day 3. And the number of dead cells is pretty high early on, right? So in day 1 in the biofilm and day 3 in the planktonic, the cells have started to die. And that's just in LB growth media, and that makes sense because after the nutrients run out, cells start to die, which happens later in the process. What I want to show is that these, when you mix Staph aureus and you incubate it with the supernatant of the biofilm, even by day 1, there is nothing there, right? So either in the biofilm or in the planktonic. If it was just the Staph aureus can't form a biofilm, then we should see a lot of staph in the supernatant or the planktonic. But we don't, right? We see almost nothing, which means that it hasn't even been a day and the supernatant has really eliminated most of the cells in there. So this suggests that there's something active going on that is killing the Staph aureus, and it is something that Burkholderia produces during biofilm growth. So we think that it is killing it. All right. So we have a lot of other data. So what happened is we found a lot of this data, and then the inhibition disappeared. And that is, we eventually figured out because we changed our bucket of LB medium. And different batches of LB medium have different sugars, different compositions, and the new one, we could not recapitulate the inhibition. So we went through a long process of trying to optimize getting this inhibition back and understanding the conditions under which we see this. We found out that glucose supplementation of the medium is really important for seeing the inhibition, but so there's a sweet spot of the right amount of glucose that you can see the inhibition, but not enough to acidify the medium. Because once you add too much sugar under these conditions, fermentation occurs and you get acid buildup. We know that other sugars are not sufficient, so galactose, sucrose, fructose, or was it lactose? I think we tested lactose. Those do not lead to the inhibition. We also know that this inhibition doesn't depend on some of the other small soluble factors that are produced by Burkholderia cenocepacia strains like pyomelanin, which is a melanin-like pigment that is thought to protect against reactive oxygen species. Ornobactin and pyocheilin, which are both siderophores, which are known to be important for harvesting iron. So we have mutants of these that do not show a difference in the inhibition. We also know that the pH, it's not a pH thing. So we can buffer this medium and we can still see the inhibition. All right. So the grant that I got is actually to figure out what is this secreted compound that kills Staph aureus and what is it made out of, and is it more than one thing? We know that Pseudomonas produces proteins, lipids, and secondary metabolites that kill Staph aureus, but we do not have the genes that make any of those in cenocepacia. So it's probably something novel. The way we go about this is quite complex. And it's one of the things that I'm working on and I need students to help with. So there are potential projects for all of you on this work. So there are physiological studies where we can further optimize the production of the inhibitory compounds. To do this, we would test different temperatures, different types of media, different batches of media. And these would be like a standard set of procedures that you'd get really good at. It involves growing up the microbes, getting them into biofilms, incubating or testing these biofilms with different variables, and then seeing if there's a dose-dependent effect. So this is really good core microbiology stuff. Another set of studies we have in the grant for this are the biochemical studies. What is the nature of the compound? Is it made out of proteins, lipids, metabolites, DNA, RNA, and other? And this is the one I'm currently working on, and I could use some help. This may involve, it's certainly going to involve the standard microbiological techniques of growing biofilms, quantitating things out of biofilms, but also doing things like protein analysis, doing biochemical treatments. That's the one I'm doing. Doing some metabolomics. I'm starting that this week. Or perhaps working with lipids, which I have no experience with. So I need some people that might be willing to learn how to work with lipids, which, believe me, are a pain in the ass because they're lipids. They're hydrophobic. So there's a whole new skill set in there that we're going to need to develop in the lab. I don't know why this is so weirdly animated. Okay, and then a genetic study. The second aim of the grant is actually to figure out what are the genetic pathways in Burkholderia that lead to the production of these compounds. And so we can do this by doing what's called a genetic screen. So if you're into genetics, this is a great one because what we do is we make random mutations in cenocepacia using existing things that we have, and then we screen them for ones that cannot kill Staph aureus. And so I have some pretty good fun ways of doing this. And then we sequence the mutants to figure out where the mutations are and what is being disrupted. And so this is a really cool genetic study that would be really complementary to the biochemical studies. We could also, eventually, if this goes well and we figure this out quickly, we can also segue into what are the genetic components in Staph aureus that are responsible for being killed. So what are the targets of the antibiotic that we think is being produced by Burkholderia? So that's a really cool one too. That's a little ways off, but if we get enough people figuring out what these molecules are, that's the next logical thing. So I am looking for a variety of students that want to work on these projects. You can see that each of these can hold 1 to 3 students. So if you're interested in these, we can talk further about how it works, or you can, we'll sit down and figure out who's going to work on what. But these are the core things. All of them will involve core microbiology. Some are more simple. So if you have a busy semester, maybe the physiological studies would be a good way to get your feet wet. Wouldn't take up too much time. The biochemical studies are going to be a little bit more, you need to be able to take the lead on learning some new techniques and reading, and will take a little bit more time. And the genetic studies are going to take a little bit more time too, because you're going to have to be picking and patching individual colonies of bacteria to do the screening. So it takes a little bit more time, right? All of them are essential for understanding this interaction, right? So they're all incredibly valuable. With that, I'm going to end the presentation, and I hope that you will now then go to the next one to learn about the other much smaller project that we have in the lab.